Okay, so we are on the hour and there's a lot to cover today. So I will start giving you all a very warm welcome to today's webinar from the International Alliance of ALS MND Associations, an update on ALS MND platform trials. For this webinar, we have captions available. If you're joining us live on Zoom, please check the chat box as I will be adding the instructions shortly, or scan the QR code on screen if you have another device available. And if you're watching the recording in our YouTube channel, you will find the instructions in the video description on how to activate the subtitles. Para este seminario web tenemos subtítulos en inglés y español disponibles. Si se unen a nosotros en vivo vía Zoom, verifiquen en la casilla del chat ya que estaré agregando las instrucciones en un momento, o pueden acceder a la traducción por medio del código QR en pantalla usando otro dispositivo. Y si están viendo la grabación en nuestro canal de YouTube, encontrarán las instrucciones en la descripción del video sobre cómo activar los subtítulos. Now I want to, oh, sorry, I skipped the wrong one. There we go. Went to the wrong. So I now want to pass the microphone to our chair for the board of directors, Tammy Moore. Tammy, the microphone is all yours.
Thanks very much, Jessica. I really wish that I could speak Spanish as fluently as you do. That was beautiful. Good morning, good afternoon, good evening, everyone. I'm Tammy Moore, and I am, in my day job, the CEO of the ALS Society of Canada. But here today, I'm here as the chair of the International Alliance. And I'm so pleased that all of you are joining us for this really important discussion. We are joined today by an incredible panel of experts who will share the latest on the ALS MND platform trials, EXPERTS-ALS, HEALEY, and SMART. The first person who we're going to have present is Professor Martin Turner. He's a professor of clinical neurology and neuroscience at Oxford University, and honorary consultant neurologist at the John Radcliffe Hospital. With over 25 years of research in ALS-MND, his work focuses on developing biomarkers, such as advancing neuroimaging and biofluid analysis to accelerate therapy discovery. He co-founded the EXPERTS-ALS biomarker-led drug screening platform and has received major awards, including fellowships from the UK Medical Research Council, the Royal College of Physicians Graham Bull Prize in Clinical Science in 2016, and the Sean M. HEALEY Prize for Innovation in ALS in 2023. He has co-authored more than 350 scientific publications and several books. The next panelist who we will have speak is Dr. Sabrina Paganoni. She is an associate professor at PM&R at Harvard Medical School Spaulding Rehabilitation Hospital. Dr. Paganoni is the co-director of the Neurological Research Institute at Massachusetts General Hospital and the Robert H. Brown Jr. Endowed Chair in Neurology. Her research focuses on clinical trials and therapy development for ALS. She is the principal investigator of several ALS clinical trials and has pioneered novel trial designs and partnership models to innovate the way investigational products are tested in ALS. She is a co-PI of the HEALEY ALS platform trial and is leading multi-center expanded access programs. Dr. Paganoni is working with other Harvard researchers on developing new assistive device technology and care models for people with ALS and other forms of motor neuron disease. Her research has been funded by NIH, nonprofits, and industry. She published more than 170 reviewed manuscripts and received several awards for her work, including the 2021 Top 10 Clinical Research Achievement Awards. She is a member of the National Academy of Medicine. And we have Professor Subhankar Pal. He is professor of neurodegenerative disorders and clinical trials and consultant neurologist at the University of Edinburgh. He is co-leader investigator of the UK-wide innovative multi-arm, multi-stage MND SMART trial, a group leader at the MRC UK Dementia Research Institute, deputy director at the Anne Rowland Clinic, and an honorary professor at the University College London and the MRC Centre of Research Excellence in Clinical Trial Innovation. His clinical and research interests are focused on improving outcomes for people with neurodegenerative disorders, including accelerating early and accurate diagnoses, evaluating atypical presentations, leading population-based disease registries for longitudinal deep clinical phenotyping, digital and wet lab biomarker development, reverse translation, and delivery of innovative clinical trials. And your moderator for today is Gethin Thomas. Gethin is currently the acting scientific director of the International Alliance of ALS MND Associations, as well as playing an advisory role and co-leading the Global MND Research Roundtable for Fight MND. Gethin has over 20 years of experience as a biomedical researcher, having published widely and secured almost $4 million in research funding. Over the last 12 years, as a senior research director, he has led a university research office, served as associate dean of research, and was the executive director of research at MND Australia. He has served on the boards of a number of organizations, including the International Alliance of ALS MND Associations. This broad experience across a spectrum of research environments and activities has enabled Gethin to develop a fundamental understanding of research strategy and operations and a deep appreciation of the challenges facing the MND research community. Now, some final instructions. Any questions you have for our panel, please write them in the Zoom question and answer box. I also want to take the opportunity to thank everyone who pre-submitted their questions. There is a lot to cover, and we will do our best to answer as many questions as we can in the time that we have. For now, I'm going to turn it over to Gethin as our moderator today.
Thank you, Tammy. Thank you for that great introduction to everyone. And as you all have seen, we have a brilliant panel here with expertise probably right across the MND ALS research spectrum. But we will ask you to keep your questions around the platform trials tonight. Otherwise, we'll run out of time very quickly if we go too broad in our questions. So we'll kick off straight away to get into the actual presentation. So first up, we have Professor Martin Turner talking to us about the EXPERT-ALS trial. All yours, Martin.
Okay, well, hopefully you can see that and hear me, and thank you very much for the invitation. So,
Dr. Turner, we're not seeing your slides, sorry.
Okay.
There we go.
How's that?
Excellent, thank you.
Good, great, thank you. Well, thank you very much for the invitation to talk about EXPERTS-ALS again, and I'm going to give you a little bit of background and then tell you where we are with things. This is a joint project between my University of Oxford and University of Sheffield, and that's with Chris McDermott, who I work very closely with. So just a reminder, what's the problem we're trying to solve? Well, you know, I've been working in this field for a long time and been involved in many trials. The way we do trials definitively at the moment, we either look at survival or we look at a rate of change in disability. And both of these are blunt instruments. They're not accurate, and they take a long time to be certain. And of course, survival is not something that we want to be reliant on. It's an extremely blunt instrument. But even disability scores, we have real problems with nuance and the variation in the patient group. Some people having much faster rates of progression. And if we do these definitive trials, these are going to require hundreds of patients. We're going to have to recruit them, usually over a year. And from the start of the idea for a study, it's going to take at least 3 to 5 years to get to the analysis. And with a median survival in ALS, as we know, of 30 months, that means that many, many people taking part in a trial will never see that result. These are the drugs that have been tried, and that represents over 30 years of work. Riluzole is the sole drug that's had a clear-cut disease-modifying effect. There's more work to be done on interleukin-2. And the dexpramipexole trial I'll highlight in the middle there, that involved nearly 1,000 patients and cost $100 million to do that negative study. This has involved 25,000, I estimate, patients over these studies. And I just don't think it's good enough, that result. But we're not short for drugs. We've got a huge number of things to test, and we simply don't have enough staff or fortunately, we don't have enough people living with ALS to test every single drug. So it's not just a money problem, and it's not just an availability problem. We've got to design the studies better. So I'm not going to talk about phase 2 trial problems, but there are problems with phase 2 trials. And as I see it, I think what we really need to do is improve the odds of winning at the phase 3 level and de-risk those studies. So the way we do that is what's called experimental medicine. We take a surrogate marker of disease activity and we see if a drug is lowering that. And if it is lowering that disease activity, then we put it back into those more definitive phase 3 studies where we can be sure that it's having a clinically meaningful effect for patients, but we can go into that with much more confidence. Now, the gift that we've been given is neurofilament light chain. It's a 30-year-old gift, and it's come to light because it's very helpful in our condition that we're interested in, ALS, and we can now measure it in the blood. It is non-specific, though. It's not a way that we diagnose the condition, but it's a marker that we can use of disease activity. And the key principles it's based on are that the amount of neurofilament light chain that you have in your blood effectively is a marker of how fast your disease is going. This is a range of diseases you can see on the graph here, but ALS comes in one of the red bars on the left as having very high levels, and that's because it's an aggressive disease, whereas Parkinson's and Alzheimer's have much lower levels on the right-hand side there because they're much slower. So this is a rate. It's not the amount of damage, it's how fast it's going. And these graphs are just showing you that level does equate in ALS to both survival, but also your rate of disability progression. So this is kind of what we're seeing. These are the different rates of progression you'll see in ALS patients, from Lou Gehrig, very typical, through to Stephen Hawking, very unusual. And over those different rates, we'd expect the NFL levels to be lower. But the key thing, and this was something we discovered in 2015, and I think was when I first began thinking about this platform, was that NFL levels stay stable for individuals throughout their condition. So if you have a high level, it's going to stay high, and if you have a low level, it's going to stay low. But that means we can predict what it's going to do, and therefore if we give you a drug and it changes, then we think that that's to do with the drug. So that's enormously powerful. And just to say, if you give these, if you give therapy to people with HIV, you give it to people with multiple sclerosis, you lower their neurofilaments, and that's associated with both obviously clinically meaningful benefit in HIV, but also clinical and MRI benefit in MS. So it's very clear that this is a surrogate marker that we can work with. And we have this extra evidence that when you give this gene therapy in SOD1 ALS, you get a very remarkable drop in the neurofilament, but the key thing is it happens a long time before you see the clinical effects, and that's what makes it so powerful. So the key thing I think that we're piloting, which is very exciting, and the FDA are very interested in using Bayesian statistics. And what this means is that we're changing our view on how we're going in the study, using a probability modeling. And so we're feeding the results into a supercomputer at intervals and then making decisions on how we're doing. This is how we do it. We take 3 drugs, we select them according to effects in the laboratory and good penetrance in the CSF. And then we randomize patients to one of those 3 drugs. They take that drug for a maximum of 6 months, and we take their blood NFL levels at various intervals. And we do a batched analysis of that, and then this regular Bayesian computer-generated futility analysis. And there's 3 outcomes. The outcome we want, of course, is the one at the bottom, but we have to accept that history would tell us that we're likely to get certainly some chances where it's just not going to do anything in the middle, or we might actually be making patients worse potentially by their neurofilaments going up. So if the model makes the call that that's happening in the first 2 scenarios, then that drug arm gets stopped. If we get a hit, then it goes in prioritized into phase 3. Now, if you ask ChatGPT to summarize what we're doing in an image, it shows this, which is actually pretty good, except the NFL screening machine is a person. It's a human, it's a person living with ALS, and that's a very key difference. We're not taking any shortcuts on safety, but the best test is in a human. Here's the 3 drugs we're trying first: metformin, ropinirole, nifedipine. These are all repurposed. They all have astonishing results in the laboratory in models of motor neurons, and we're very excited that they represent potential to improve patients, which we're testing to see what it does to neurofilament. And we've also got a partnership with Raya Therapeutic, which we're pleased to announce, and they're bringing an industry novel compound to the table, never been tried in ALS. It's safe to use in humans, and that's the direction we want to go. We want to use the best repurposed drugs, but we also want new drugs from pharma, and we're excited to be working with Raya. Here's the update. We've randomized 112 people in just over a year. I always want more than we've got, but that's enough to enable us to start doing our first futility analyses, because the model is powered to pick up a change in as little as 30 patients per group. We opened 10 sites initially, and we're opening a lot more so that we can really accelerate the recruitment. Just a reminder that we are doing some biomarker work, so all the samples get collected, so we can look at exceptions within each group and see people who are doing different things to different drugs, and also develop new biomarkers. So my last slide is really just saying that we're changing the conversation, really, and that's what I want to do. We will improve the chances of success, both in platforms you'll hear about later, the HEALEY platform and MND Smart. I think if we can show a surrogate marker of disease activity reduction first. The human is the best place to develop human surrogate markers, not animal models. And experimental medicine, just to be clear, is not experimenting in terms of safety, but it's just using the human to develop these markers. We don't know what the key pathways are in ALS, so just a reminder that repurposed drugs are not second best. There are many, many examples of where repurposed drugs have been highly effective, and that's because they're having off-target effects that we don't expect. Industry's coming, which is great, and we're keen to get lots of conversations going about helping them to de-risk their compounds so that they don't invest $100 million if we haven't had that early human signal. And we were, working with data that's published from the dexpranopexole study. I think that study would have been called using our model within 6 months, and would have been probably at 1% of the actual cost. So this could make a huge difference. My hope is that every single person diagnosed with ALS at the point of their diagnosis will be asked if they'd like to help the greater good and help everyone by taking part in a drug screen for the first 6 months. And if we did that with every single patient, we would be pouring in drugs with really good chances of success into the excellent platforms that you're going to hear about next. So a big shout out to the huge team involved in doing this, both at Sheffield and in Oxford, and the fabulous funding that we've had from NIHR, but also the charities, MND Association, My Name's Dotty, and LifeArc. So thank you very much.
Thank you, Marty. So what we'll do is we'll save all the questions for the end, and we'll run a panel Q&A session. So if you have burning questions, please pop them in the chat, and we'll be sure to present them at the end of the webinar. So the next speaker, we have Dr. Sabrina Paganoni, who is going to tell us all about the updates to the HEALEY platform. Take it away, please, Dr. Paganoni.
Thank you so much. It's so wonderful to be here. I just want to acknowledge the audience. I see text messages in the chat, people connecting from around the globe. This is wonderful. So thank you so much to the Alliance for bringing us together. As I heard the excellent presentation from Professor Turner, I see how all of us here are doing, are designing our trials slightly different, but they're all complementary. They all have the same goal, which is to bring new drugs, new treatments to people living with ALS as fast as possible. And we want to do it, again, around the globe to provide research options to everyone, regardless of where you live. And so listening to 3 different trials today, one after the other, is really heartwarming. It really shows how the clinical research community is thinking creatively and in an innovative way to make the drug development process faster. So I'll present my experience. I've been working on this trial for a few years now. I'm the co-principal investigator together with Dr. Sukovic. And really, this is a large trial, the HEALEY ALS platform trial, that's grounded in a fantastic collaboration within so many in the ALS community. So I just want to start by highlighting the role of our academic partners, many industry partners, and so many patient groups and foundations that have come together, again, because we all recognize the importance of moving fast. When we set up the trial, when we designed the trial in 2020, and still today, we have a fantastic group of patient advisors. And they, Sandy, one of our advisors, put it very powerfully when we started this journey a few years ago. She reminded us that people living with ALS don't work on the human time clock. Their clock, the ALS clock, is faster. And so she and Mr. HEALEY, and obviously the name is the trial is named after Mr. HEALEY, who also challenged us to work on the ALS clock. Mr. HEALEY was a brilliant businessman from the Boston area, and he realized that the business of clinical trials was too slow. And that's why he challenged to think outside the box, learn from other disease areas, and set up a trial that would be more efficient. And that's exactly what we did. So in 2020, we opened the HEALEY ALS platform trial after learning about it from our colleagues in oncology and some other neurological diseases. And we realized that platform trials have several advantages over traditional trials because we can cut the development time significantly, we can reduce costs, and most importantly, we can reduce the number of people assigned to placebo, and we can talk more about it. I'll touch on some of these topics during the talk. Obviously, we'll have more time during the Q&A. But essentially, 5 years after we launched the trial, I can tell you that we have data and evidence that all of this is exactly what's happening. So we're very excited. Again, we started the trial in 2020, inspired by our patients and really as a partnership with so many in the community. And now we're seeing the results. And I'm happy to share them with you today. So platform trials are indeed a win for all stakeholders. We've been working again with the patient community. And importantly, also, we've been working with regulators, the FDA in our case, since we are located in the US. Many clinical trial sites, again, US-based, for this particular trial, but I'm so happy that there are other platform trials in other regions. And we've been working closely with many industry partners and investigators. The basic principle of every platform trial is that we have a shared infrastructure. So rather than reinventing the wheel every time, we create an opportunity, a home for every person living with ALS, and an opportunity for them to participate in research, hopefully right at diagnosis. As Professor Turner just mentioned, we want to give opportunities to everyone who has ALS to contribute to research, to have access to an experimental drug, and also help the community. In our trial, we call each drug a regimen. That's the nomenclature that we are using. And so far, we have completed the testing of 7 regimens, and we are testing number 8 right now, with more to come. So, so far, again, we have completed testing of 7 regimens. You can see them listed here. I'm not going to go into the details of these because of time constraints. I do want to mention that all our papers, all our manuscripts are available open access. So you can find them online. All the details have been, fully disclosed and published, and at least for the first 5 regimens, and number 6 and 7 are under revision right now. So again, we're committed to sharing results as quickly as possible. And we're very excited that of the first 7 drugs that we tested, 2 are moving to phase 3 trial. And essentially, our trial is also a trial to prioritize drugs. So the main goal of the platform trial is to screen drugs rapidly to identify the most promising to go into phase 3 for definitive testing. The design is slightly different from the previous trial, but essentially the concept is the same. We want to provide a rapid infrastructure to efficiently screen drugs and move only the most promising ones to phase 3. And again, of the first 7, 2 out of 7, it's 28%. So I would say, a significant result here in terms of prioritizing drugs and being able to move some of these drugs to the next phase of testing. Our trial is placebo-controlled, but then everyone who completes the trial is given the opportunity to take the drug long-term. And during the placebo-controlled period of the trial, we have a favorable active to placebo ratio of 3:1 because we can share the same placebo group. And so essentially we're minimizing the number of the number of people allocated to placebo. We're maximizing the number of people that receive active drug from the start, and then everyone can get active drug long-term in the active treatment extension. Throughout participation, we pay close attention to biomarkers such as neurofilament levels, a variety of other biomarkers. Again, I will be happy to go more in details into the details of these in the Q&A. But essentially, we do deep genotyping, deep phenotyping, extensive biomarker characterization for every single participant, so that then we can learn from this collective experience. And while we are progressing drugs to phase 3, we're also learning from each and every participant. We're offering the same options, the same opportunities. All these drugs are available at every participating site. And right now we have over 80 sites that are active in the US, and they are part of the NILS consortium. It's wonderful that every region has its own consortium of clinical sites. In the US, it's called NILS, the Network of Excellence for ALS, and 80 sites are participating. Our idea is to give the same opportunities to people, regardless of whether they live on the East Coast or West Coast or in the middle of the country. We want the trial to be available as close as possible to where people live, so that as soon as possible after diagnosis, they can enroll. Just to kind of briefly summarize the results of the first 5 years, I just want to highlight that, again, we have completed tested of 7 drugs, and we have included 1,300 participants, really great collaboration with hundreds of people working on this trial. Importantly, we have collected thousands of biosamples, and I'll go back to that in a second, but again, want to highlight the importance of biomarker characterization here to move drugs to the next phase of testing. And right now we're enrolling for regimen number 8. That's actively enrolling right now as part of this trial. And also we are committed to communication. So again, we published all our papers, open access 10 publications so far, including the results of the first 5 regimens, as well as other publications about other aspects of the trial, such as the operational efficiencies, the scientific efficiencies, with many other publications in the pipeline. And we have engaged with many other disease groups. It's really wonderful to see that many people want to learn from the experience of platform trials that have been successfully launched. And so we're working with many disease areas, both within the neurosciences and outside of neurology, to help other disease areas set up their own platform trials, because now, again, both investigators and industry recognize the power of this approach. I want to go back to biomarkers for a second. Now, we have a small but important placebo group, as I mentioned earlier. This placebo group essentially serves as a natural history study embedded within the trial. So we can learn a lot from the people who had a short placebo experience before going to active treatment within our trial. That's important because there's still a lot that we don't know about ALS. Obviously, we're very excited about neurofilament levels and about other emerging biomarkers, but we also recognize that people who participate in research want their experience to be as useful as possible. And so everyone who participated, especially in the placebo group in this trial, their experience is really amplified and their impact is maximized, because this natural history study is giving us literally thousands of samples as well as data points that have now been retired or essentially archived and are available for open science. So we are very happy to work with researchers around the globe to mine the data, do more experiments on these samples that are informing our understanding of the science behind ALS. Another aspect of this trial that's very unique is that the trial is an adaptive trial. So the trial can learn from its own experience. As I said earlier, we opened the platform, the infrastructure in 2020, and we have continued to learn from it. In fact, based on analysis of the results of the first 5 regimens, the first 5 drugs, we have actually revised the protocol. And so the new regimen that's enrolling right now, regimen number 8, is enrolling under a new and improved master protocol. So the protocol itself has leveraged all the learnings from the previous regimens, including tweaks in sort of study design, eligibility criteria, more flexibility for remote visits, a lot of user-friendly features and new biomarkers that have been added. So right now we're very excited to enroll for regimen I, or the Horizon regimen. This is a new drug, again, another industry partner, very happy to work with Horizon. The drug targets autophagy. The goal here is to clear TDP-43 accumulation in ALS, and I'm also very happy to work with the co-lead investigators, Drs. Berry and Weiss. For every regimen, we work closely with experts in different areas of ALS science, such as biomarkers and other aspects. I'm very happy again to work with Drs. Berry and Weiss. So if you're interested, I know today is really a short presentation, we'll have time for Q&A, but I do want to say that we are trying to be present for people living with ALS to the best of our abilities. So we have a central patient navigator, Miss Catherine Small. Many of you may already know her. She's available to contact, to talk to people. She's basically a central resource for navigation to help people living with ALS access trials, especially this trial. She's available by phone or email. We also have an online presence on different social media, and the QR codes here will take you to our website. Especially we do regular webinars. We have a newsletter. So if you want to get in information, again, please feel free to use these QR codes. Feel free to also contact me and also Dr. Sukovic, the PI of the trial, and Miss Drake, who is our senior director, and we are happy to receive feedback from the community. We also accept applications from industry on a rolling basis. We are continuing to grow the trial, and we are excited to partner with everyone in the community who would like to offer this option to, again, accelerate drug development in ALS. So once again, thank you so much for your attention. And now I guess I'll turn it over to the moderator, and I look forward to the Q&A segment of this presentation. Thank you.
Thank you, Dr. Paganoni. That was a fantastic overview of the HEALEY platform trial. So we'll just move straight along on to Professor Sivanka Pal, who will take us through the SMART MND SMART trial.
Thank you very much, Gethin. Can you hear me and see me okay?
Yep, no problem. That all looks good.
Perfect. Just like to echo the previous speakers really and thank the Alliance for the invitation to speak. It's always wonderful to take part in these webinars, sharing the progress that we're making to improve outcomes for people with MND ALS globally. I'll start off just with an introduction to our team. It's a large interdisciplinary and collaborative team. Really what we're trying to do is take state-of-the-art approaches in drug discovery now through to improving outcomes for people using innovative trial designs for definitive and rapid testing. And this obviously involves a collaboration between drug discovery scientists, experts in clinical trial methodology, trialists and neurologists, but crucially, people living with MND ALS are at the heart of everything that we're doing. One of the reasons that we're particularly excited and being at a tipping point now is that we understand a lot more about the underlying biology of what's driving MND ALS. We understand the complex multicellular nature of the disease. There are many cell types affected, and therefore also many targets for therapeutic intervention. We know a lot more about the protein that's misfolding in this condition and the way that the nerve cells are affected. And the approaches that we're taking to select the best medicines to test in people is really taking state-of-the-art approaches across these different domains. I'll talk about high-throughput screening assays that use human-based stem cell models. There's obviously a huge explosion in AI and machine learning computational approaches in drug screening, thinking about all of the literature that's emerging all of the time and how we keep on top of that with systematic reviews, and improving animal models. So if you think about the stem cell screens that we have, so our group have been looking at combining human-based stem cell screens with high-throughput drug screening of libraries that are FDA and EMA approved. And using these approaches, you can screen thousands of drugs at pace and look at the nature of these different protein misfolding disorders that occur. So TDP-43 is the abnormal protein that we know is implicated in motor neuron disease, and you can replicate what goes on in people in the stem cell model in a dish, expose that to drugs and see how the overall clinical picture improves. And similarly, you can look at how the supporting nerve cells, the astrocytes and microglia, are affected by drugs. And if we screen these drugs at pace, what you find is that some of these drugs have a hit on these assays, but not just one or two of the assays, some have hits across multiple assays. And you can find classes of drugs that show benefit. This is particularly exciting. It also opens the route to combination therapies, which we're particularly interested in, drugs that might have an advantage to reduce the protein misfolding and other drugs that might have a benefit on the supporting cells. And maybe if you test them together, much in the way that is tested in cancer medicine or in infectious diseases, there might be a better outcome for people. You can refine the findings by testing these drugs in more sophisticated animal models. This is just an example of a mouse model that has the TDP43 misfolded protein aggregated in the brain, and you can expose these mice to the drugs and see what the outcomes are in terms of their pathology. And it's using these approaches that we've selected the drugs that we've tested in MND Smart so far. And an example is tacrolimus, a drug that we're testing at the moment, a repurposed medicine that has been used for immunosuppression in other disorders. But this has compelling data from our human discovery platforms in reducing the misfolded protein and dampening down the effect on the supporting cells, the astrocytes, and the inflammation of the microglia. It also had a positive benefit on the mouse model. And we've heard from the previous talks about the benefits of the multi-arm, multi-stage adaptive trial design. If we were testing these drugs that are coming through at pace now from the discovery platforms, individually in series, each drug against a placebo, it would take the best part of 4 to 5 years to identify the drug, to then write the protocol, to then collect the data, clean the data and report on the data. And just to test 4 drugs in this way in series would take the best part of 2 decades. And we've heard about testing drugs simultaneously against a contemporaneous or a standard of care arm that's running at the same time. It has efficiencies in time, in cost, in the number of people required, and therefore the burden to people living with this difficult disease as well. We also are an adaptive trial design, which means that new drugs can be introduced at any point, and we're looking at all of the accumulating data as it's emerging to make decisions about whether a drug should stop or continue. So we also launched in 2020. In the last 6 years, this is a summary of what we've been doing. We've tested 3 drugs definitively now, memantine, trazodone and amantadine, which came through the drug selection that I mentioned. We're looking at the overall outcome of survival, which is approved by the regulators globally. We're looking at the interim analysis points at functioning and the biomarker neurofilament light chain that you've heard about. And we're also a phase 3 trial. So the early stages of this trial might be akin to a phase 2 study, but as we go on into interim stages 2 and 3 with more participants followed up for longer duration, we have definitive results that would meet the regulatory standards of being a phase 3 trial. We're looking at other outcome measures like people's mood and the effect on memory and thinking, and also on their breathing. The platform, much as you've heard from the previous study, the HEALEY trial, we're learning as we go along, and we've designed a flexible platform with a master protocol and appendices, and we have very inclusive entry criteria, and then we look at the populations individually. So for example, ALS, the commonest form of MND, is the population that will focus on the analysis, but we do include people with other subtypes of MND as well. We have drugs with different routes of administration, different dosing schedules, different safety monitoring requirements, and all of this is possible because of the flexible study design. Again, we've been very successful with recruitment. We've had to build infrastructure that didn't really exist before when we started. We now have 26 sites across the whole of the UK. What this graph really shows to me is the unmet need still amongst people living with MND-ALS to participate and the real desire to take part in studies to improve outcomes. I mentioned we've tested 3 drugs definitively in half the time it would have taken if these were serial 2-arm study designs. And really, again, as you've heard before, what we're doing is striving for equity of access to people, regardless of ethnicity, background in terms of deprivation, and thinking about generalizable results for the population as a whole. It's important when we have definitive results to really share these with the community, the patient groups and the scientific community quickly, and we've managed to do that for the closed treatment arms. What's important, again, is that these are definitive results. These are studies of several hundred people. The first 2 drugs were tested over 170 people on the active treatment arms. I mentioned a generalizable population. We had very high rates of data integrity. Another historical criticism of studies has been the high rate of dropout and missing data, and we haven't really experienced that in this study so far. Very recently, we had definitive results for amantadine, and this was tested again in over 170 people. On this occasion, we included neurofilament light chain data, again, refining the study as new evidence comes along. The next drug that we're testing is a drug called montelukast. This is a drug that's, again, come through our high-throughput screens. This was a primary candidate in an assay called the excitotoxicity assay, and also it modulated activity of astrocytes, the supporting nerve cells. This is a drug that's been tested before in other neurodegenerative disorders like Alzheimer's and Parkinson's, so we know that it crosses the blood-brain barrier, we know that it's safe, and we know the dose to start with in people living with ALS. So our protocol is being implemented and going through regulatory approvals this year. And we have a suite of other drugs for which we need to think about answering questions around, do they cross the blood-brain barrier? What is the optimal dose in people living with ALS? And are they safe in this vulnerable population? And we're planning a suite of experimental medicine studies in a small number of people studied in detail before taking them through to the definitive platform. I mentioned that people living with MND-ALS have been involved in every stage of the study design, really from the thinking of the study protocol all the way through to the dissemination and authorship of outputs. And that's been one of the real reasons we feel we've been successful since we launched. We've heard as well about the amazing gains that can occur now that we have several thousands of samples from hundreds of participants over many, many years of follow-up. And this allows us to really interrogate and look at the new biomarkers that are emerging, high sensitivity biomarkers, established approaches, but also new approaches that look at proteins in the blood. And one example of this is looking at an assay called ELISA, which is a very high sensitive blood-based assay that can look at a panel of over 120 proteins in people with MND compared to controls. And this is from our baseline population in the arms that have now closed. And in addition to neurofilament light chain, there are a whole range of other proteins that are elevated in people living with MND. And what's interesting now is to study the change over time and also the response to treatment in these individuals. Now, we're very excited about the possibility of decentralized trials, so assessing people more in the community powered by digital technologies. It will obviously democratize access to trials for people, reduce burden and improve data integrity. And an area that we've been focusing on are digital measures like speech, and we very much hope to incorporate these in the study in the very near future. We heard about the benefits that come from people having access to participating in trials, and what has become apparent to us is there's a big unmet need for a platform trial in Europe, and we've been working closely with partners in the QRND for ALS network in France, Germany and Belgium, and we're pleased that we're working very closely with the Paris Brain Institute to open MND Smart in Paris and then across the network in France over the next year. So in summary, MND Smart is really a large interdisciplinary team ranging from state-of-the-art drug selection methodology all the way through to innovative testing in people. And our forward momentum is to test many more drugs that come through the pipeline, including combination therapies and adopting experimental medicine approaches, decentralizing the trials using digital assessments, and expanding into Europe, collaborating with existing networks. So many, many people to thank across multiple different areas that they're all listed here. And thank you in particular to our funders who are listed as well, and very much looking forward to the questions. Thanks very much.
Thank you, Professor Pal, for a fantastic overview, and thank you to all our speakers for really setting the scene of where we are with platform trials. So we've got a few questions lined up already from the audience, so I'll just, I think most of them are applicable to all three of you, so it'd be great to hear the different perspectives from the different trials. So the first one is, I think you've all mentioned that platform trials are allowed to have perhaps broader selection criteria. inclusion criteria for participants in the trials. Could you just sort of discuss that? Is that the case, or are you still trying to have a fairly homogeneous population like we do with perhaps single treatment trials? Savanka, do you want to start since you already
I can talk about the approach that we've taken in MND Smart, which is essentially, if we do develop treatments and interventions that are successful, we ultimately want them to work across the whole population of people living with MND, ALS, and the subtypes. And so therefore we need to demonstrate that these drugs are effective and safe across the whole population. So we have very broad inclusion criteria. We also understand, though, that some of these biomarkers, particularly the functional rating scales, may not change at the same rate in different populations. And so the way we've done that, or addressed that, is to look at a primary analysis population that focuses on those individuals who are changing quickly enough for there to be a measure that we could look at the interim analyses, but to look at survival as the overall outcome of the study, which can be looked at in any individual with any subtype of disease. And then obviously when you're recruiting several hundred participants, you could do a sub-analyses on different subtypes of people with MND at the end of the study. And so if you pre-specify at the beginning and say, right, I'm going to look at the whole study population, but I'm also going to look at these individual subtypes, and this is the number of people I need to have in the study to be able to do that properly, that allows you to look at things in detail that way.
Right. Thank you. Dr. Paganoni, would you like to comment?
Yeah, and I agree with Professor Paul. That's definitely one possible approach. In our case, we use the slightly different approach, which is to have a specific set of criteria for enrollment in the platform trial, but we've also offered something called expanded access for people who are not eligible for the trial itself. So again, all of these different designs, have all pros and cons, and again, there's not necessarily just one approach. One approach, again, as was just mentioned, is to pre-specify the population of interest. In our case, we basically run two separate but parallel studies, the actual trial and then an expanded access for people that don't qualify for the trial. At least we've been able to do that for, so far, 4 out of 7 of the drugs.
And Professor Turner?
Yeah, well, we have quite a broad entry criteria for experts. Everyone's getting a drug, of course, so we do have a kind of responsibility to make sure that everyone's data is going to be used in the model. So we can admit about 75% of all new diagnoses, and the other 25% are in a group whose disease is moving very much more slowly, which is good for them, but it means that they're likely to have very low neurofilament levels, and so for our modelling, it wouldn't be useful. Now, that doesn't mean, when we get a result through that we wouldn't roll it out for those patients, of course. But there is just a slightly narrower criteria because we want to make sure the whole point is to get an answer very quickly. But it's still 75% of patients can come in.
Great, thank you. On a different topic, how do you actually select the sort of candidate treatments you're testing in your on the platforms? Are there specific criteria are more important than others? Martin, sorry.
Yeah, well, I think there's several principles really. One is it's actually quite a basic one, is sort of an independent group. So we do have experts, and we've involved through links through LifeArc, the Medicines Catapult, and industry experts really on how you pick preclinical data. I mean, there's a lot of debate in the field about what the criteria should be for preclinical data, and I'd like to see that harmonized. I was discussing that at a meeting in ALS with ALS Canada just at the weekend. I mean, that would be good that we all agreed internationally what the key preclinical data are. But roughly speaking, there are a number of pathways that are clearly of priority. We want CNS penetrant drugs. But I think people are moving away, if I might say, from some of reliance on mouse data. It's not the SOD1 mouse has been extremely good for genetic SOD1 ALS, but it has great limitations really more widely. So I think you've heard about some in silico developments from Savanka. But I think we perhaps need to get a bit more harmonization internationally on how we do this.
Dr. Paganani, you comment on selecting?
Yeah, no, I agree with Professor Turner. Absolutely. This is complicated because we don't have a gold standard and only one approach to be able to predict from basic science data what would be successful. I would say, now that I worked in this field for 15 years, and, it's really a lesson in humility in terms of, really trying to learn from all the data. In my mind, it's more the totality of the data, not any just one specific model or one specific experiment, but it's really the totality of the data. In terms of how we select the drugs for the platform trial, we've been very fortunate to have lots of great discussions with industry. We've taken the approach to, partner with industry to test novel compounds. And so far, through different workshops and industry interactions, we have had, I believe, 52 applications from companies so far. And it's really conversation with companies to make sure that we understand whether their compound is the right fit for the platform trial at that time. That's based on solid preclinical data as well as readiness to be in the trial. And that's a combination of things such as, for example, availability of a biomarker, availability of a target engagement readout, ability to penetrate into the brain, previous human data, obviously phases, studies that have to progress from small initial studies with some initial safety. These are all new compounds. So we do need some previous human experience to make sure that the drugs are safe. And so once we look at the entire package, we can partner with companies and move it to the platform trial. I would say there's been companies that we started working with, and then, different things happen in terms of, the need maybe for more data or additional studies. So again, this is a very much dynamic and evolving situation because we're all trying to partner to do what's best for patients and only put in drugs that are the best fit at that particular time, knowing that some drugs we may be able to get back to once additional data has been accumulated.
And Professor Paul?
Yes, so I agree with everything that's been said. It's an exciting time because of the pipeline of drugs that's coming through, and there are various different approaches, and we heard about the totality of evidence being quite broad now. Involving people living with the condition has been very important to us as well, so they have helped with the drug prioritization, thinking about the complexities as well of routes of administration, dosing schedules, requirements for safety monitoring. So that's been helpful in terms of what they think. But clearly we need to make sure that the drug is getting to where we think it should be getting to. Does it cross the blood-brain barrier? That's been mentioned a couple of times. And is it safe in this vulnerable population? We don't want to be causing more harm to people. And we've heard about some of the measures that we can use now to see if a drug is working or not, the functional measures, the survival, the neurofilament light chain, but we often need a marker of target engagement. Is the drug hitting the target? And I think we're all agreed that that needs to improve as well. So many factors, but still an exciting time because of the pipeline being very much ahead of where it was maybe a decade or two ago.
Thank you. Professor Paul, you mentioned that you're looking to expand the trial overseas into Europe. Can I ask the same question of experts and HEALEY trials, whether there are opportunities for other countries to be involved and to expand them beyond the UK and the US?
I'm happy to start. Yeah, absolutely. We would love to expand to other regions. The reason we started in the US is because, as I said earlier, when we were thinking about setting up the platform trial in 2019, we learned a lot from other colleagues, some other neurological diseases, but also especially cancer, because cancer and infectious diseases have had many other successful platform trials. And so we learned a lot from them. And one good piece of advice we received was to first build the infrastructure and then expand it. Because, this is a kind of a complex infrastructure. It's definitely doable, but you do need to put a lot of upfront investment and care in terms of, fleshing out all the details and the different aspects. So our colleagues told us, well, start in one region with one regulatory authority. So the FDA in our case, or obviously there's a different regulatory authority in every region. And so that's what we did. And I think that was good advice. And now that the infrastructure is really such a well-oiled machine, almost autopilot at this point, now we can expand. We're having some of these conversations. So again, it will all depend on sort of some of the logistics, making sure that we have all the right partners and funding and all of that. But definitely that's a good idea. Having said that, I don't know that we need necessarily just one platform trial. I will say that I always love having these webinars, learning from others and different approaches. Again, they're very much complementary. ALS is a complex disease, and to think that there's only one way to approach this is really naive. We need all the best people in the world to think creatively, innovate, and learn from one another like we're doing today. So I would say, while it's important to continue to give options, and definitely it's good for every trial, I think, to expand beyond one country, I would say that it's also very good to have different networks and learn from one another as we each try to grow and give more opportunities to patients.
Yeah. I mean, I don't think I've got anything more to add really to what Sabrina said. Certainly, experts is something that several countries are interested in setting up their own networks, and the individual components are not hard really. So I think it's something that if we're showing that it's as efficient as I think it can be, I think it'll be a very obvious thing for countries to set this up as a regular thing. So yeah, no barriers to that.
So perhaps on a more complex note, then, do you think there'll be opportunities in the future for, say, the Global South to be involved in these trials as well? What do you say?
I think so. There's definitely a willingness amongst researchers, trialists, clinicians, and an unmet need amongst people. I think what you've heard is it's more the barriers are regulatory and financial, possibly. They're the sort of things that slow things down. But certainly for other platform trials and other disease areas, they have shown that's been possible in the Global South. And as we've heard before, we just need to make sure that we've bedded in and are successful in what we're doing and know what we're doing and then can expand that incrementally. So what we've achieved has been over a short period of time in our home nations, but now it's probably right to extend.
Thank you. I have a specific question about the EXPERTS-ALS. People in Italy are very keen on hearing the outcomes from your first 3 drugs. Do you have a timeline of when do you think you'll have?
Well, it all depends on the model. It's kind of like the all-seeing eye, really. So it will be doing the first futility analysis next month. So we put the data in that we've got, and the computer says, "Yep, I can make a call on this arm, or this arm, or no, I need more time." So we'll just have to see. That's the sort of testing part of it. But the statistical power of the model is that call could be made with as few as 28 patients who've all taken a given drug for 6 months each. So who knows? We'll have to see. I think what we're hoping really is that ideally we'd be able to screen 3 to 6 drugs a year. That's really the hope. So yeah, exciting over the next few months to see what comes out.
That'd be brilliant. Also, I think all 3 of you mentioned that lived experience input has been critical in helping develop and evolve the trials. Could you sort of talk a little bit more about that and how useful it has been, and has it actually resulted in changes into how you design or have evolved the platforms?
I'm happy to start with that one. So people living with MND-ALS have been central to everything that we've done, really, from the study design to delivery. It's a tough condition to be living with 24/7, and taking part in clinical trials is a commitment and a burden often. And so actually minimizing that burden for people really does require their input in how we think about delivery. So I've mentioned a few areas. They've been involved in the regulatory submissions, that's to the ethics, and in the UK, the MHRA. They've been involved in thinking about the inclusion criteria, thinking also about the drugs that we've selected, the routes of administration, the safety monitoring requirements, and then actually been really helpful in applications for funding as well. So thinking about co-production that way, and obviously in the final outputs of studies, we've had co-authorship with our PPI group. So it's been a very rewarding experience for us working closely with our patient group.
Yeah, I'm happy to share our experience, which is similar to Professor Paul's experience. In our case, we have a couple of groups. One is called the Patient Advisory Group, and then we have something called the Recruitment Retention Engagement Committee. So those groups meet on a regular basis, several times a year, and they include people with lived experience, patients, caregivers, families, and they really helped us design the trial. In fact, we started meeting even before the trial started enrolling in 2019, and we continue to meet to this day with new people joining. And and in addition to kind of reviewing the design and providing input and sharing information about, things that would be helpful in terms of the design, I just want to give an example that I still remember very vividly. When we launched the trial, that was exactly when the US went into the lockdown for COVID. That was early 2020. And at the time, obviously, things like Zoom or Teams or this type of video conferences were available, but they were not as common as today. I know it sounds like incredible to think about it, but there were only few webinars or few things like this, meetings like today's meeting, available for people living with ALS. And so it was Sandy, one of our patient advisors, who during one of those early meetings in early 2020, asked us to do a weekly webinar for patients to be up to date on the progress of the platform trial. Because at the time, think about it, like February, March 2020, everyone was thinking about COVID, but people living with ALS also wanted very much the trial to get up and running. And so that was kind of an example of something I would have not thought about, quite frankly, because, at the time, even I thought, who would want to see us? Who would want to see a webinar, watch a webinar every week? That was very much something I would have never thought about. And it was the patient community who asked us to do that. And so we've done that. We've had a weekly webinar for many years. And only last year, I think now there are so many other webinars, which is wonderful. Other groups doing so many webinars for ALS. Now we only have it twice a month. But that has really been our way to keep the community posted on which sites are available, which drugs are available, how many people are enrolling, how many spots are left, because we understand the importance of transparent communication and also live updates. It's very hard to just follow publications. I mean, again, we have open access publications, but those are only kind of, they only come out every few months. So that was one example. I just wanted to provide this concrete example to say that including the patient community perspective makes the trial better and makes us think in directions that, quite frankly, I would have never thought. And I'm so grateful for that input.
Thank you. Professor Turner, you have the last word.
Yeah, well, we're doing a big qualitative piece with experts, which involves, asking people on their experience, really, including why patients sometimes drop out, and no surprises, but it's a reminder that we have to address the simple things like the burden of travel for these studies. That has to be reasonable. But there's also new things. We're giving everybody a drug. And so actually the psychological components are slightly different. If people are told they're going to get side effects or what the side effects might be, then you risk something called the nocebo effect, where you actually induce more side effects whereas in your placebo control trial, people might be quite, in some ways, pleased to have a side effect, because then that means that they're on the drug arm. So there's lots of aspects, actually, that I think I'm learning, as always, over many years. Everything I'm learning is from what the patients are telling us. So we're doing a big piece to kind of understand how this particular design, which is new, fits into the community.
Thank you. And it's fantastic to hear that the lived experience voice is so strong, and I think that's just increasing in all aspects of MND ALS research around the world. So we've reached the end of our time slot, and I'd just like to thank speakers yet again. It's been absolutely brilliant. I think you've given us a fantastic overview and hopefully some real hope for people living with this horrible disease around the world. So thank you again, and I'd like to thank all the listeners, everyone who's dialed in and asked some very insightful questions. So good night to everyone, and I think there'll be a recording available pretty soon in the next day or two. So for those, if you have people in your networks who've missed it, please make sure they know it's available, so as many people as possible can listen to this brilliant information. So good night, or good night from me anyway. So for the rest of you, enjoy your day. Thank you.
Thank you.
Thank you.